Hello my dear aspirants, I hope you all are doing well. Today we will be discussing recombinant DNA technology taking help of blue-white screening method. So I hope this class will be useful for you. This particular topic is required in CSIR NET, Northeast Slet as well as Neet and class 12 boards. So watch the class till end and just comment below if you don't understand any point. If you like my videos then please subscribe to my channel. I just want to provide an additional information to the students of Northeast Slet that if you want to take coaching from me, if you want to learn the detailed syllabus for Slet, Northeast Slet, then you can just comment down below or you can contact to the given number. Okay, so let us start about recombinant DNA technology, which can be also known as gene cloning. So here I have written some major points about what recombinant DNA technology is. In the next page, we will explain how these things work. So the first thing you have to know is a fragment of DNA containing the gene to be cloned is inserted in a circular DNA molecule known as vector to produce recombinant DNA molecule. So I hope you all know what is a vector. Okay, I hope you all know what is a vector. Vector is nothing but imagine it to be a vehicle. Okay, what you do, vector can be a taxi, okay, a bus, train, any vehicle which can carry passengers or which can carry any goods. So vector is such a vehicle, molecular vehicle you can call. There are different types of vectors. Let me take example of plasmid vector. Today I will not go in depth what vector is. Here I will just focus on recombinant DNA technology, considering that you all know properly what vector is. Imagine we have a plasmid vector. Okay, plasmid is nothing but a DNA, okay, a circular DNA which is present in bacteria, additional DNA which is present in bacteria. It is circular and it is double stranded. Plasmid vector, it is present where? It is present in bacteria, okay, and it has the self-replicating property. Okay, so now this plasmid vector, here a gene will be inserted. What type of gene will be inserted? A gene of our interest. Suppose I have a container. Okay, I want to send this container to this destination. This destination will be called a host, and this container which I want to send here will be called gene. Okay, just an analogy, nothing else. So if I want to send this container to this particular destination, I can't directly send it. I will need one vehicle which can be a car. You can just relate things like that. So first what I have to do, I have to insert this gene into this vector. I have to put the container inside a car, and then only this particular car has the capacity to go inside the destination. So this is how vector works. We all know that insulin is a very important hormone of our body. So that particular insulin gene will be present in our DNA only. Suppose this is our DNA. Okay, suppose this is our DNA, and this particular region is suppose the gene that codes for insulin. Okay, this is the gene for insulin. Imagine we need lots of insulin, but our body is producing only a little amount of insulin. I'm just giving an example. Okay, so if you need a lots of insulin, that means you need a lots of cell that will contain this particular gene or this region. So if I want to make lots of copy of this particular gene, it is naturally not that possible. So we used to do recombinant DNA technology so that we get many copies of only this gene we are concerned about. So first what we will do, we will isolate this gene. Okay, at first we will isolate this gene, and this gene is nothing but the gene of insulin. Okay, now if we isolate this particular gene, then can we get copies without replication? No, right? We will require replication. Lots of replication has to be occurred so that we get lots of copy of this insulin. First step done. First step, we have isolated the insulin gene. Second step, what we'll do? Suppose here we had a bacteria, and this bacteria had one plasmid. Okay, then we will isolate the plasmid like this. Okay, we will isolate the plasmid. we have first isolated the insulin gene, and this is our gene of interest. Okay, we are already interested in this particular gene. Then we'll isolate the plasmid also from a bacteria. Okay, from one bacteria we'll isolate a plasmid, bacteria A. Okay, so we have isolated the plasmid also. Okay, so in this test tube, the cutting part was done by treating with restriction endonuclease in both the test tube. So we got cut pieces of the gene, and we have got the cut pieces of the plasmid. Then we'll mix both of them. We'll mix both of them in one test tube like this, and now we will add ligase enzyme. Okay, ligase, the role of ligase enzyme is to ligate or is to join. So there will be some plasmid which the role of ligase enzyme is just to ligate, just to join. So the ligase enzyme, there is probability that the ligase enzyme can join the cut pieces of the vector also like this, or maybe because these things were mixed all together, or the ligase enzyme can mix them also, can join our gene with the plasmid also. So this particular thing will be called recombinant, which we need actually. This is our requirement. And these particular plasmid which have done self ligation will be called self ligated, because ligase enzyme's role is just to join. It does not recognize it does not know that it has to join recombinant one or self ligated one or whatever. So here we will get two types of things. One, the plasmid will join to each other's cut ends, or sometimes the plasmid can join with the our gene of interest also. So when the plasmid will join, okay, whenever we will get this type of molecule, this type of DNA molecule, then this will be called recombinant DNA molecule. I hope you understood why it is called recombinant or hybrid, because it has our gene of interest, which is this particular red colored gene is the insulin gene, and this green colored thing is the plasmid. Okay, so here we have got recombinant. So now how we can recognize, how we can distinguish? We will insert these things into host bacteria. Okay, there is possibility that the recombinant one can insert the host, and there is also a possibility that the self-ligated one can also insert the host bacteria. Okay, so three possibilities will be there. One, when none of them will enter the host, then it will be called non-transformed. Okay, nothing is inserted in the host, it will be called non-transformed. When any of them will enter, then it will be called transformed. Okay, because in the host bacteria at least we have something, then it will be called transformed. Again, this one will be also called transformed because at least some or any one of them has inserted. When the perfect one will occur, that means when the recombinant molecule will enter the host, then it will be called transformed recombinant. Okay, transformed and also recombinant because it has the recombinant molecule. And if the self-ligated one will enter, then it will be called transformed but non-recombinant. Okay. Now which one out of A, B and C, our requirement is only B, because B is transformed also, recombinant also. So we will need B. How we can eliminate A and C? Because these things are not visible by naked eye. So here I will discuss about blue-white screening, which is a method to identify the recombinant and transformed bacteria. So here just remember one thing, the plasmid of the bacteria, they have particular regions like ampicillin resistance, some antibiotic resistance gene, and also some enzymatic activity. Okay, some enzymatic genes present. So here the plasmid, if it has a LAC-Z, okay, a LAC-Z gene, it produces one protein or enzyme known as beta-galactosidase. Okay, LAC-Z gene of the plasmid, it can produce beta-galactosidase, and beta-galactosidase helps in producing blue colonies. Helps in producing blue colonies whenever we place them in a Petri plate. So remember one thing, if recombination will occur, takes place, then this gene is inserted right at the LAC-Z area. So imagine suppose till here it is a leg jet gene, and if the gene is inserting right here, then the leg jet will be disrupted. Okay, the leg jet will be functionless. So as a result, the leg jet cannot produce beta galactosidase and there will be no blue colonies. So just remember, whenever there will be recombination, then the recombination of our gene of interest will take place in the leg jet region here, and whenever a gene will insert in this region, then insertional inactivation will occur. That means the gene of this area will be functionless because the gene will be broken. Okay, you can remember like that, the gene will be broken, so leg jet will not be functional anymore. So this is the step 1, where we have put ampicillin, which is an antibiotic, and we have placed all the bacteria. Here I am just giving example of one bacteria, but here there may be many bacteria. Okay, this ampicillin resistant gene of the plasmid, it provides resistance to ampicillin. This particular gene was not hampered, so whenever we will place the bacteria in ampicillin containing medium, then only the transformed bacteria, that is B and C, will be able to grow, because B and C bacteria has the plasmid. The plasmid had ampicillin resistance, which provide resistance towards ampicillin. So this A bacteria is not transformed, neither it is recombinant, so this will be eliminated, because here we have put ampicillin antibiotic, and this A bacteria will not know how to survive in ampicillin because it is lacking ampicillin resistance gene, because it has no plasmid in it. Next, the B bacteria, it has the plasmid which has the ampicillin resistance gene, so it can survive in ampicillin. Third bacteria, it has the plasmid, and the plasmid has ampicillin resistance gene in it, so the third bacteria will be also able to survive in ampicillin media. Okay, so now we have B and C, but we are concerned only about B. We only need B. We have to eliminate C also. So for that we have to do one step 2, which is the blue-white screening. So here we have cell B and C. A was eliminated in the first step itself. So here we have put X-gal, which is a chromogenic substance. Okay, beta galactosidase can convert X-gal into a blue color. Okay, so in B and C we have put X-gal. So in case of C only, beta galactosidase will be able to convert X-gal into blue color. Why? Because here the entire plasmid is intact. The entire plasmid is intact and its leg jet region was not hampered. But in case of the recombinant one, here in this particular region it had the leg jet gene, which was inactivated after insertion of our gene of interest. So here white colonies will be formed because the leg jet is now functionless. And why leg jet gene is functionless? Because the gene of our interest, that is the insulin gene, will be inserted here, and here there will be blue colonies. Why? Because the leg jet gene was as it is. It was not hampered by any insertion of any gene. So as a result, where we will get blue colored colonies will be eliminated, those cells will be eliminated, and where we will get white colonies will be selected. So now we got our B bacteria or the B host cells, which are our desired cells. After this B bacteria, they will divide, there will be cell division. Okay, the bacteria, because it can divide in a very rapid manner, very fast manner, so there will be lots of cells. Okay, lots of cell within a short period of time, and with the increasing progeny, increasing generations, our gene of interest will also keep on replicating, so we will get a lot of our gene of interest. Okay, so this is how cloning will take place. Now our final aim was to increase the copy of our gene of interest, and as the bacteria will divide, along with their cell, our gene of interest will also divide, because this plasmid will do replication, this entire plasmid will do replication, and as a result our gene of interest, which was recombined with the plasmid, will also do replication, and thus we will get many copies of our gene of interest. Okay, I hope you understood today's class. Thank you, and please subscribe to my channel.